huge pleasure to be able to give a talk to the Barcelona Collaboratorium for Modeling and Predictive Biology. My talk is under the rather audacious title, Can Modeling Predict Life? Or is it the case that harnessing stochasticity, chance, is the key to understanding life and its intelligence? Is life therefore not perfectly predictable? And if so, why? I start by drawing attention to relevant publications. This book, The Pacemaker Channels of the Heart, is an account of my modeling work on the heart over the last 62 to 65 years. And that's been published this month and is available on Amazon. There are two articles highlighted in this slide too: How the Central Dogma and the Theory of Selfish Genes Misled Evolutionary and Medical Sciences in Understanding Multifactorial Diseases, and a related article in the Journal of Physiology, The Cardiac Pacemakers, a Paradigm of Robustness in Evolutionary Biology. My first part, for the first 15 minutes, will be to ask the question, how did stochastic robustness evolve? And that will also help us to explain why association scores do not reveal causation. How robustness evolved in the heart was the focus of a paper my team published way back in 1992 in an article in a journal of the Royal Society. And what we showed there is in the case of the pacemaker mechanism of the heart, at least 4 genetic components, or rather their protein output from those genetic sequences, enable the networks, the physiological functional networks that generate the heartbeat, to be functioning. And any one of those is sufficient for pacemaker activity to occur reliably. Each can substitute for the others, therefore. And that means that only one of them is completely sufficient. What that means, of course, is each is a backup for the others. That creates the great robustness of the heart pacemaker. And that was shown by taking a model that is based on detailed experimental work on the single sinus nodes of a rabbit heart, revealing the automatic voltage rhythm that you see there on the right in the tracing. That's a model simulation of what in practice happens also in a natural sinus node when you record from it experimentally. But you'll notice there we're carrying out an experiment on it, and that experiment is fully described in the next slide. The top trace shows what I showed in the previous slide. The frequency starts relatively high, and then it just modestly changes during those 10 seconds of activity. And the way we produce the change in rhythm was this. 2 of those 4 channel mechanisms that I outlined in the previous slide are shown here. One is a background sodium channel at the bottom, and in the middle, a sodium-potassium mixed ion channel coded for by HCN genes. And what we did in the model experiment was comparable to what we could also do in the actual experiments, which is to reduce the main current, in this case it was the background sodium channel, by first 20%, then 60%, 40%, 20%, and then finally complete knockout. Now, if that was the only mechanism generating the rhythm would stop. You'd have an association score of 100%. Instead, you get an association score of around 10 to 15%. And the reason is clear from what the model shows, because as that component was being reduced and eventually completely removed, the other mechanism, the HCN mechanism coding for the hyperpolarizing activated mixed inward current, that simply replaces the functional process needed to keep the pacemaker rhythm going. The system has replacement of function by another mechanism and is therefore completely robust. There are 4 mechanisms, each of which can substitute for one or other. I asked the question, therefore, when we made this discovery in 1992, how widespread is robustness of this kind? And I later found that, in fact, over 90% of physiological functions are extremely robust. That was first shown in a systematic study of the 6,000 genes in the organism yeast, done by Hillenmeyer and his colleagues in 2008. And what they showed with knockouts of most of those genes is that 80% of the gene knockouts are actually silent. The association score is 0 in normal physiological conditions. But they then went on to show that when you remove key nutritive elements from the chemistry of the environment in which the yeast is living, each of nearly all of those silent mechanisms gets revealed. There is therefore massive robustness in the metabolism and reproduction processes of yeast. Those are the 2 processes being measured. Now, what that means is that the great majority of gene association scores are going to be small. Robustness will ensure that. If the robustness is perfectly good, the association score will be 0. If it's strong but not perfect, it'll be a small association score. That explains why, in the output of the Human Genome Project, the great majority of association scores are unexpectedly very low. So low, in fact, that what people have done is to add together all the association scores. For example, if you take a disease like schizophrenia, you will find around 300 genes that are associated to some degree or another, modest amount of association with that disease. They just add the scores together to form the polygenic scores. This study, published just 2 years ago in 2023 in the British Medical Journal Medicine, shows that even that is not predictive. To quote from their study, polygenic risk scores perform poorly in population screening, in individual risk prediction, and in population risk stratification. And they conclude strong claims about the effect of polygenic risk scores on healthcare seem to be disproportionate to their performance. That is a major shock, because we were promised that within 10 years of human genome sequencing, that would be 2010, cancer, heart disease, schizophrenia, dementia would all be susceptible to genetic cures. Where are those cures? 25 years later. I think, therefore, in concluding this part of my talk, that it really is time to completely replace the central dogma of molecular biology. Crick's formulation of it is shown on the left in that slide. DNA enabling RNAs to be made, enabling proteins to be made, and no back arrows that can take it the other way. Well, indeed there aren't. At that level of organization of a biological system, that process doesn't exist. Yet we know it must. Because when a new virus invaded our bodies during the pandemic, our immune systems were working overtime to inform the immune system cells, please, by chance, find as many new DNA sequences for immunoglobulins to grab the virus as you can. And the system then selected which cells had managed to get the answer. They were asked to produce many new immunoglobulin molecules, while the others were told to recycle them. So my conclusion from this part of the talk is that DNA is simply not the blueprint for life. And the key elements of the argument is that, first of all, DNA does not even begin to have the necessary properties. It's a very poor self-replicator. It relies on the cell, the living cell, to generate an accurate process of replication of DNA before the cell can divide. And indeed, chemically, DNA itself is a very passive molecule. The real active molecules in living systems are the RNAs, which can form modestly strong enzymes, and of course the proteins that form extremely strong enzymes. And the other major conclusion from this part of my talk is that association scores simply do not prove causation. The YouTube link there is to a lecture I gave in Santiago de Chile earlier this year on just that topic. I come to the second part of my talk, which explains how organisms harness the stochasticity of their water-based milieu in all the cells of the body. Water is in fact a very unusual chemical, particularly the range over which it is liquid, because the individual atoms, hydrogen and oxygen, would be frozen at very, very much lower temperatures than 0 degrees centigrade. So that's the first major fact. Second is that let's look at the smallest living organism, a bacterium. This is Salmonella as a bacterium. It's around 1 by 4 microns in size. It contains 20 billion water molecules. Now, a lot of those are bound and therefore not completely free, but a lot are. And they will be showing the typical Brownian motion of water particles in a liquid medium. No available computer, not available to us today anyway, could possibly reproduce at a molecular level all of those 20 billion water molecules, or whatever fraction of them are free to move around. Yet this organism survives brilliantly well. It can swim around with those flagella to find its food, its intelligence, if you like to call it that, in a tiny cellular volume. People are attributing intelligence now to artificial systems. This can do much better than most artificial systems. If we attribute intelligence to an artificial system, then I think we have to attribute it even to a tiny organism like a bacteria. So what do organisms do with our stochasticity? Well, they use it, as the example from the immune system showed. Because to take DNA replication, the standard view is that it replicates like a crystal. That's why the self-replication arrows in Crick's diagram are solid. You'll notice that in my diagram, I made them dotted to show that it's only partially a self-replicator. Because when unwound, the double helix bases will naturally attract their pairs, A with T, C with G. And in a purely chemical environment, this works well up to about 1 in 10,000 base pairs. Human genome of 3 billion base pairs would have hundreds of thousands of errors. And this makes stochasticity massively available to all living cells. How do they use it? Well, what they do is they use it intelligently. First of all, living cells themselves, with their army of cut and paste enzymes, correct virtually all of those errors. The cell doesn't allow itself to divide until that criteria is reached. Accurate copying, therefore, replication, is not independent of the living cell. But when required, cells can then use that stochasticity to create new DNA. It's precisely what the immune system did during the pandemic with pinpoint accuracy, just allowing one part of the sequence for an immunotherapy to change. And then the immune system chooses which cells have achieved that particular target. Other systems in living organisms, like the nervous system, can also use massive stochasticity, which has been measured in nervous systems in all species. And I finish with quoting, first of all, the godfather of artificial intelligence. 75 years ago, Alan Turing wrote this seminal paper in 1950 in the philosophical journal Mind. And he wrote, "It is probably wise to include a random element in a learning machine. A random element is rather useful when we're searching for a solution of some problem. And since there's probably a very large number of satisfactory solutions, the random method seems to be a better one than the systematic." And he went on to point out, and it should be noticed, that it is used in the process of evolution. That's the godfather of artificial intelligence, Alan Turing. My conclusions, therefore, are living organisms precisely control the openness in their stochasticity. Each organizational level achieves that by harnessing the stochasticity of the level below. This precise multi-level meshing leading to purposive creative behavior. But once harnessed, randomness is no longer random, it's directional. Meaning making, therefore, arises from the harnessing, and all living organisms can do that. And that leaves the system open to those improbable, possibly rarely occurring forms of behavior when they are required. So my 21st century challenge to the modeling of living systems is this: how can the multi-level directional selection of stochasticity be best incorporated into our models. Because we need good modeling of how, for example, the immune and nervous systems achieve this. They must be harnessing stochasticity. And I finish with thanks to the co-author of the book, Raymond Noble, Understanding Living Systems, published just 2 years ago by Cambridge University Press. You'll find many of the arguments I presented today on replication of DNA and harnessing of stochasticity in that little book. And just published this month, available on Amazon, is The Pacemaker Channels of the Heart, which is an account of my work and my team's work over a period of 65 years, all the way from the first heart cell model in 1960 to the situation today in 2025. That includes many of the arguments I presented very briefly here today on the future of modeling work in living systems. I also thank Ren Bourret, a colleague in Paris, for many discussions and comments on what I presented today. Thank you very much, and I wish all good fortune to the modelers of the future. Goodbye, and thank you.